And then we move on to the next speaker, Alexandre Mayran. Alexandre is working at the Federal School of Technology in Lausanne, Switzerland, the EPFL.
Bonjour.
And is going to continue telling us about gastruloids.
While this loads, first of all, I'd like to thank Denis for inviting me to present with you today. And yeah, as Ben said, I think it's a great lineup, and I'm super interested to see the rest of the day. So here, basically, I'm showing you a small movie that we acquired recently in the laboratory. And so I'm a postdoctoral fellow in the laboratory of Denis, actually. And he welcomed me in the lab, and I've been working on actually very similar topics that Ben just presented, so it's going to make my job at the introduction much easier than it would have been. I'm interested in the self-organization, and I'm using these pseudo-embryos, these gastruloids that he quickly presented, as a model to study these processes. And so here you can see two of these pseudo-embryonic structures, one that manages to self-organize, forming from this disorganized fashion a polarized structure, which then gets slowly restricted into a band pattern. The gene that is marked in green is called MESP1, and I'll come back to this. And on the right, you will see one that actually fails to elongate. And so what are the processes involved in successful versus unsuccessful self-organization? And so as Ben presented, here I'm showing these drawings from Haeckel, which actually took this observation from von Baer, as this stage, which is called the phylotypic stage, where the embryo tends to look very similar to one another from species that in the endpoint look very different. And so indeed, this contrasted with the early step of development, where the embryo actually differs quite a lot between species. And so Denis in 1994 proposed this hourglass model where constraint on the mechanism, namely in what he proposed, it was the collinearity of Hox's genes, makes some kind of constraint that forces the embryo to undergo a very similar morphology, which will then diverge when species gets different. And so Lewis Wolpert had this very nice quote that actually is very time relevant, because my brother is actually getting married in 2 days, so please don't tell him I'm reading this quote. But he said that not birth, not marriage, not death, gastrulation is the most important time in your life, and gastrulation is the process that allows the embryos that have very different starting morphologies to converge into a conserved body plan. And so if you take the human or the mouse embryo, as you can see at this stage, they look quite different. Yet, when you go to early post-gastrulation stage, you have a morphology that is quite similar between these two. And this is the process of gastrulation, where a portion of the epiblast cells will start migrating. And so gastrulation is all about cell movement, the activation of the different transcriptional program of the germ layers. And this occurs in different ways in different species. But you have these cells that begins to migrate through the embryo, forming the different ectoderm, mesoderm, and endodermal germ layers. And so now we have new tools that allows us to reinvestigate these processes. And here you can see a mouse embryo that's been cultivated in vitro. And you'll see, I guess, more of in vitro culture of embryos later on during the day. But you can see how the cell migrate. You can trace back, based on their final location, their initial location. And so you can start to question how this whole process occurs in time by rewinding it and going forward. And so other technical advancement consist in the use of these artificial embryos, which are derived from stem cells. And so in 2014, the laboratory of Alfonso with Susanna van den Brink developed this gastruloid model and went on with further characterization, including in this collaboration with Denis Duboule and Matthias Lutolf. And so the gastruloids have been mentioned by Ben in the previous presentation, but what they're interested for is that they are able to recapitulate gene expression patterns that are typically found in an embryo with quite limited positional information intake. So the idea is that you cultivate this aggregate of cells on which you put a chiron molecule, which is a Wnt activator. It represses the Wnt inhibitor GSK3 beta in the media. So you have a signal coming from all across, and yet these structures are able to form a single axe and elongate and form complex or more complex gene expression pattern. They can form mesodermal structures, a neuronal or endodermal. They can form multiple axes, as you can see with these anterior-posterior restricted genes, dorsoventral ones, mediolateral, so they can become quite complex. And the laboratory of Denis Duboule actually became interested in this structure because they are able to recapitulate the nested expression of host genes. And you can see that this nested expression of host genes also has a temporal collinearity where the order of the Hox, in this case the Hox A cluster, occurs progressively in time. And so gastruloids can also be expanded, and I'm sure we'll hear more about that later with Jesse. But in this paper from Giuliana Rossi, you can see that you can push gastruloids to form more complex beating heart structures. The laboratory of Alexander Van Oudenaarden with Susanna van den Brink in collaboration with Alfonso and Jesse Van Vliet showed that they can form complex structures with a proper or more proper than in the original gastruloids. So mites and neural tubes, and I don't want to spoil too much of his presentation. So I'll just, basically the idea is that gastruloids are able to form complex structure, and we can extend their potential, and other examples of that have been shown before. And alternative structure will probably be presented later today also by Christine T, so I'm super excited about these presentations. But so the questions I wanted to ask when I started in Denis' lab was, how do these aggregates that are spheres of originally what used to be homogeneous population of embryonic stem cells, when subjected to this homogeneous signal, can form symmetry breaking, can break symmetry morphologically and elongate along a single axe. And so this is what I define in this presentation as self-organization. And so is this process linked to sulfate differentiation? And when is this diversification of cell identity that we observe initiated? So we first did a single cell experiment time course in order to identify the first time where we detect transcriptional diversity. And so when you look at the earliest time point that we've tested at 48 hours, you can see the identity of the cell is quite homogeneous transcriptionally. But if you go 1 day later, so after you've subjected the aggregates to chiron, you can now see that there's been diversification between this population that resemble primitive streak identity and this nascent mesoderm identity. And later on, now the gastruloid cells have diverged into neuronal, somatics, and endodermal cells. But so if we could focus on this first time point where we detect transcriptional diversity, and we looked at the program that is specific to each portion, we can see all these genes that are preferentially expressed in this nascent mesoderm population, but we don't know their localization. Are they polarized? What about their spatial organization? And so Dominique, when she started in the lab, she performed some in situ hybridization of these markers, and we could see that actually there's these transcriptional program seems to be very disorganized. It's spotted in a salt and pepper-like pattern, and so we could not say from this that at this stage, the initial transcriptional diversification occurs in already self-organized fashion. This seems to occur later. And so when we follow this event in time, and here she performed, Dominique performed hybridization chain reaction through time on gastruloids, you can see the same thing at 72 hours, but 1 day later, it's now organized into this band-like pattern. And if you go even later on, you can still see this band structure. So what Dominique did then is that she generated a reporter lines to be able to perform live imaging of this process. And so she inserted GFP with a nuclear localization signal into the MESP1 gene, and this allowed us to get an expression that resembled very strongly the expression of MESP1. So with the help of Pierre Ostoy and Ariane Berkowski in the laboratory of NDOTS, we managed to generate this movie, which I showed you in the first slide, which now allows us to assess really the transition between these two, which seems to be quite critical in the process of self-organization. And hopefully next time I can actually give you the conclusions of this experiment. But so, since we detected that at 72 hours is the initial stage where we detect transcriptional diversification, we wondered, did we miss anything? Is there any pre-existing information, maybe at the protein level, maybe at the signaling level, that we could not see when we did this single-cell experiment, single-cell RNA-seq experiment? So what we went on to do is that we dissociated the gastruloid at different stages. So here we've dissociated at 48 hours, here at 72 hours. And when you look at these, although the structures are smaller because in the process you lose some cells, they're able to elongate quite nicely. So they're completely, maybe not completely, but because we didn't comprehensively test this, but they seem to be quite resilient to loss of positional information that dissociation brings. But when you do it a bit later on, at this stage when elongation has started, you can now see that they're not able to reform these elongated structures. And if you go just slightly before, when it's still spherical, but and not yet asymmetrical on a morphological level, now you can see they're completely fine. And so all this is quantified here. When you take around 96 hours, which are still round, they're able to self-organize completely. But the ones that have changed morphology already, it's too late. You can't disrupt the positional information and reaggregate them to have a nice elongation. So, as I mentioned in the first part, in the introduction, gastrulation is characterized by the emergence of cell movement. And so this is often seen as an EMT, which is characterized in part by the switch from E-cadherin, which is expressed in epithelial cells, to N-cadherin, which is expressed in mesenchymal cells. And so when you look at the transition from early gastruloids here to late gastruloid here, you can see that E-cadherin is switched off, while N-cadherin is switched on. And so this stage is actually when things seems to be started. And if you so we focused on this because this seems to be the transition point between these two epithelial to mesenchymal status. And so here what we did is that we looked at the gene-specific velocity. So basically what we do here is that we look at the splicing patterns of genes in order to assess whether the gene is being actively transcribed or transcription has stopped. And so in red, you will see genes that have stopped transcribing actively, and in blue is when you have new transcription occurring. And so when you look at E-cadherin, you can see this portion, this small portion, which is actually the one expressing the nascent mesoderm gene in a program which I mentioned earlier, have begin repressing E-cadherin, while a large chunk of the 72-hour starts to transcribe N-cadherin. And so if we look a bit more closely in time, this time in bulk, you can now see that the repression of E-cadherin is mainly visible at 84 level on the stable transcript, but then it keeps decreasing. while the induction of N-cadherin begins as early as 72 hours, and it reaches its maximum at 84. So from this, it looks like the induction of N-cadherin precedes the repression of E-cadherin. But we wanted to test that and look at it spatially. So with the help of Maxine, we performed some immunofluorescence of E-cadherin and N-cadherin during gastruloids development. And so as you can see, and as expected early on, we only have E-cadherin expression. And later on, we have most cells expressing N-cadherin protein. But if you take this 96-hour time period, where at the transcriptional level we detect very limited amount of RNA, we still have some protein of E-cadherin, and it's restricted to a small portion of the gastruloids, while the remaining part do not have E-cadherin expression, while the whole structure express N-cadherin. So this small chunk of gastruloids co-express both markers. And so if you try to go a bit more precise in time, you can see that the induction of N-cadherin begins in what seems like sporadically in a few variable locations in the gastruloids. But when you go at this time point, you can now see that most cells co-express both E and N-cadherin in the gastruloids. But so this tells us that indeed we can see that the activation of N-cadherin occurs before the repression of E-cadherin. But this doesn't tell us anything about the identity of the cells that do the switch. And so what Maxine tested then is to try to connect this process with SOX2 used in later stages as a neuronal marker and TBX6 as a marker of mesodermal, of presemitic mesodermal cell, I should say. And so you can see that SOX2 early on is expressed because of its role in the epiblast, and then it gets repressed. But then it gets turned on again, and you can see that at 96 hours, the cells that express SOX2 also express E-cadherin, while the cells that expressed TBX6 do not express E-cadherin. So this suggests that the repression of E-cadherin occurs preferentially at this stage in the mesodermal cells. But if you go later on, you can see that SOX2 positive cells have also repressed E-cadherin. So both cell identities repress E-cadherin expression, but they do it at a different time. And so if you go a bit more precisely, you can see the initiation of TBX6, which does not correlate with loss of E-cadherin. So this would suggest that the activation of this program occurs before the repression of E-cadherin. But both neuronal and somatic lineages repress it, they just do it at different time. But so this is mostly correlation, and this doesn't tell us anything about the role of this. So we tried to assess functionally whether that was important. So the first thing we did is we tested whether N-cadherin, which is also called CDH2, is important for the self-organization process. So the hypothesis being that maybe cells that start expressing it can move around gastruloid and find each other. And so we went out and used CRISPR-Cas9 to delete N-cadherin, sorry. And so when we grew gastruloids out of ES cells that lacked N-cadherin, actually we can see that they elongate quite nicely, even in better in a way than the typical gastruloids that we grow. So clearly, the self-organization into a single elongating gastruloid does not require N-cadherin. When you look, although you have these still quite important morphological changes, when you look transcriptionally, you can see that very little is happening. A typical view in the field is that constraints and forces can impact the transcription of gene, while here, clearly we would expect that we've impacted the internal forces by affecting the way cells interact. We have more elongated structure, so in a way, gradients can be altered, but we have very little that changed transcriptionally. When we look at the anterior-posterior organization of this structure, so we used HOXB9 as a marker of the posterior end, you can see that they're properly patterned. You have this negative portion and this posterior HOXB9 expressing portion. So N-cadherin function is dispensable for gastruloid self-organization. And so what we went on to test then is whether the repression of icadarin that occurs here is important. And so one of the main actors of this repression during gastrulation is through the action of snail. And so as you can see in gastruloids, snail expressing cells at 72 hours are the same ones that have turned off icadarin expression. And so in order to prevent the repression of icadarin, the approach we took was indirect. We removed snail to remove the repressor of icadarin. And so what this is shown here, we removed the first exon of snail 1, and when we now grew gastruloids out of ES cells missing snail 1 expression, we could see very variable phenotypes. And so the most frequent one being multiple weak elongation. Sometimes they were semi-properly formed or polarized, and sometimes the elongation was extremely weak or they remained ovoid. And so when we now looked transcriptionally at what happened, we could see that a lot more was happening transcriptionally. Namely, the most upregulated gene is icadarin, which is consistent with its known role as repressor of icadarin, but a lot more events are happening. Namely, the somatic program seems to be downregulated. And when we look now at anterior-posterior organization in this structure, so here we used MeOX1 as a marker of the anterior portion of gastruloid, and OXB9 as a marker of the posterior. But when you stain snail mutant with these two markers, you can see that sometimes MeOX1 is expressed in the central portions of gastruloid, sometimes it's in protrusions, sometimes it's properly patterned. And for OXB9, you see the same thing, sometimes in the protrusion, sometimes in the central body. So what this suggests is that intrinsically, each of these cells know what they're supposed to be. They're just not able to convert this information that they have intrinsically into the global organization of the gastruloids. And so we finalized the characterization of this mutant to try to really assess and dissect what was happening at the cell identity level. And so we performed the single cell experiment time course of the snail mutant. And so the whole data set is represented here, where you can see the wild type cells here and the mutant cells here. And so we did that at 3 different stages. First, at 72 hours. So what you can see is that the 72-hour gastruloid looks like this. You have this portion with this extension, which corresponds to the nascent mesoderm. And so you can see that this population is absent from the snail mutant. So they're not able to form this cell identity at that stage, at least. When you go slightly later on, at 96 hours, you can see that normally the cells would have committed to either neuronal or somatic identities. But in the snail mutant, you have these uncommitted cells that, in theory, would still be free to go into one or the other identity. And when you go later on, you can see that the mesodermal fate, which normally is quite well represented in these 120-hour gastruloids, is now very weakly represented and late. You don't see these cells that are more advanced in the differentiation process. And in contrast, the neuronal lineage is actually more advanced in the mutant than it should be. So in the wild type, we see a majority of what could be characterized as NMPs, while we have more advanced differentiated neurons in the mutant. So it looks like the timing and the pace of differentiation is completely altered. It's not that everything is late. Some lineage are too early, some lineages are too late, as if they don't really know what to do anymore. And so, I don't know how yet I'm on time, but today I hope I've shown you that gastruloid cell can diversify from a homogeneous population. The initial diversification occurs at multiple variable locations, suggesting the process could be stochastic, but as Ben said, it's very difficult to show randomness in biology. There is a cell-specific coordination of the timing of EMT, meaning it occurs in all lineages, or both neuronal and somatic at least, but at different time. The completion of EMT is crucial for gastruloid self-organization, and the EMT regulator, SNAIL1, can regulate the cell-specific timing of completion. And so with this, I'd like to acknowledge all the people of the lab. So first of all, Denis, who allowed me to work on this project in his lab, all the lab members, which I actually mentioned, I think all of them, except Lucille, who helped with the analysis, and Catherine, who helped with generating cell lines, Ariane in the oats lab, all the other collaborators who I didn't talk about, I didn't mention their collaborations in this presentation, Viventis for help with the microscope for the light sheet that you saw in the first slide, and all the funding agency and the EPFL platform. And I thank you for your attention and take any questions.